Super here?
Yeah, sure. So we'll go on as well. Is that right?
Yeah, sure.
Okay, we'll get started. We're running a little bit late. So I'll just introduce Dr. Tyrosine from all the way here from University of Saskatchewan in Saskatoon. There's a vet college there, in case some of you don't know that. Most of you probably will. And
Good afternoon. I'm very excited to be here, and a big thank to the organizing team of this wonderful symposium to give me the opportunity to come here and share some of the data and information with you. Today I'm going to talk about F-receptor tyrosine kinases and their potential for development of targeted cancer therapies for both humans and companion animals. So I divided my talk today in 3 sections. The first section I'm going to talk about some research data, these data coming from human cancer research side, which I spent most of my previous time in that area, and then I'm going to talk about comparative oncology and how those experiences and research data could be utilized in a comparative oncology. So I had the opportunity to work with different models of human cancer research, including leukemia, colon cancer, prostate cancer, and breast cancer. For today, I'm going just to focus on the work on breast cancer because it has more relevance or clinical relevance in terms of using DNA damaging drugs, the small molecule inhibitors, and synthetic antibodies. So let me introduce the F-receptor tyrosine kinases. F-receptor tyrosine kinases are the biggest subgroup of receptor tyrosine kinases. They are divided into 2 groups, FA and FB. There are 9 receptors in FA group and 5 in FB group. They are expressed at cell membrane. The difference between FA and FB groups are the type of ligands that they bind to. FA receptors, they bind to ephrin A ligands, and FB receptors in cell-cell interactions. So why F-receptor tyrosine kinases are important in cancer research? If you look at their function, they regulate cell proliferation, morphology, adhesion, movement, survival, and differentiation. These are all important functions that either being altered or needed by cancer cells. And indeed, abnormal alterations of F receptors have been detected in multiple human malignancies. So when we talk about alteration, what type of alteration are we talking about? I'm going to talk about 2 main conditions. Either we have decreased expression or function of receptor, or we have increased expression or function of a receptor. And I'm going to talk about 3 different scenarios, how to deal with this condition to have some suggestions for clinical implications. In the first case, when we have decreased expression, if that decrease is partial, we can plan therapy based on the function, based on the remaining function of the F receptors, or even by enhancing that function. And if the decrease is complete, or that reduction is complete, it's a complete elimination of the receptor, we can detect lethal pairs with that target molecule, which I'm going to expand on that. And in the case that we have increased expression or function, we can design synthetic antibodies to interfere with those cancer-promoting functions of the receptor. So let's start with the first scenario. Expression of F receptors is increased. Most of the time, they are increased in multiple human malignancies. There is one receptor, FB6, which its expression is consistently reduced compared to the normal tissue. So that raises the question that what aspects of function of FP6 is not desirable or not welcome by the tumor, so they want to get rid of it. We try to answer that question. As I mentioned, in general, F receptor in FB receptor expression is reduced in cancer compared to normal tissue. However, if you look at this panel, the FB6 receptor expression is better preserved in triple-negative breast cancer tumors. So there's still some function remaining in triple-negative breast cancer compared to other subtypes of breast cancer. So we built our model, research models on triple-negative breast cancer. The cell lines that we used for our investigations were MDA-MB231. In this cell line, FB6 promoter is methylated and there's no expression of FB6. So we restored FB6 expression in this cell line. BT20 and HCC70, they have endogenous expression of FB6, so we use shRNA to silence FB6 expression. So for our experimentation, FB6 increases cell-cell contacts. These are MDA-231 cells. You see two colonies here. Each of these colonies developed or grew from a single cell. So in control cells, you see how the colony looks like. Their cells are elongated. They invade to the surrounding of the colony. There are lots of space between the cells. However, when we restored FB6, that increased cell-cell contacts and they form these kind of closed colonies. We also test motility of the cells in regards to FB6 presence, and we found that FB6 reduces cell motility. This is a scratch or wound healing assay. So basically, it's a monolayer of cells. We make this a scratch, and we monitor how fast cell can come into the scratch and close it. And you see after restoration of FB6, even at 36 hours, there is still significant gap left compared to control, which was, the gap was completely closed. So if FB6 increases cell-cell contacts and reduces cell motility, can it reduce tumor invasiveness? So we wanted to test that. We injected MDA cells with or without FB6 into mouse. We found very curious observation there. If you could look at here, the tumors that had restored FB6, they had greater growth rate and faster development compared to the controls, especially at the initial time points after injection. So what could cause that? Why those tumors are growing faster? We look at the angiogenesis in the tumor, there wasn't any difference in blood vessel formation in those tumors. There was another possible answer for that, which I'm going to talk about it. So, and it means that not all type of cells that we have in population are able or capable of developing tumor when injected into a mice. There is basically, or in fact, there is just a little or small subpopulation of cells that are capable of developing a new tumor. We call them tumor-initiating cells. So we are seeing that increased growth rate, initial growth rate, when FB6 is restored. Could that be due to increased proliferation of tumor-initiating cells? To test that, we cultured these cells in the presence of FB6 or absence of FB6 in tumor spheres. Tumor sphere is a specific culture condition that favors only growth of tumor-initiating cells. And by doing that, we observed that, yes, indeed, FB6 increased proliferation of tumor-initiating cells. This is very interesting because tumor-initiating cells usually stay in a dormant, less proliferative state. That's one of the reasons they are responsible for drug resistance, especially when DNA-damaging drugs are used, like doxorubicin. So because they are not proliferating very fast, they're still there and they can cause tumor relapse. So if FB6 kind of pushes them out of their dormancy or less low proliferative state, can it make them more sensitive to DNA-damaging drugs? So we tested this hypothesis in animal model. Here is a PDX model that we used. And you see tumors that had FB6, they had higher growth rates. They developed faster or bigger. However, they had more profound sensitivity to doxorubicin compared to their controls. In fact, if you look at this graph, in the presence of FB6, we had about 80% reduction in tumor size when tumors treated with doxorubicin. And in control, we had only 10%. So what's the clinical message from here? We could recommend that DNA damaging drugs are better choices for treating tumors that have higher levels of FB6 expression. So let's see and talk about this next scenario when we have complete elimination of a receptor. So we need to find synthetic lethal pairs in that situation. But what is that? So there is a concept of synthetic lethality. The idea is that the function of two genes are somehow related and they are necessary for cell survival. When both of them are intact, the cells are in good shape. And when one of the genes are eliminated or suppressed, the other gene can compensate for the lack of that gene. And again, there is no adverse effect on the cells. However, when both of them are eliminated or suppressed, then that is a lethal combination for the cells. So in tumors that eliminate FB6 for any reason that they don't want it, we can find what other genes are lethal pairs with FB6 and then try to target those in order to have a lethal effect on cancer cells. So we did a screening to find those lethal pairs with FB6. It needs a condition of FB6 plus or minus, and then we silenced up to 18,000 different genes individually in both conditions. We do high throughput screening, and from that, we found actually 113 potential lethal pairs with FB6. It means that in the condition that FB6 is absent, suppression or inhibition either of these genes individually can be lethal for the cells among those 113 other factors. So basically, our research shows that in tumors that eliminate FB6, if we use Src kinase inhibitors, such as KX2391, we should see more profound effect on the cells in terms of inhibition compared to the condition that FB6 is present. And we did that experiment. We treated cells without FB6 or with FB6 with Src kinase inhibitor, and as we hypothesized, we had bigger inhibition when FB6 was absent. So what is the clinical message from this experiment or this project? We found that FB6 and Src are a synthetic lethal pair. So this means for tumors that don't have FB6, Src inhibitors could be a good or possible option for treatment. However, in tumors that maintain FB6, the use of Src kinase inhibitors may not be very effective because the remaining function of FB6 can compensate for that inhibition. So KX2 actually is a drug, FDA-approved drug, which is in clinical trials for breast cancer, responsible for that resistance development. And last, when we have increased expression or function of F receptors, we can target that with synthetic antibodies. This project has been done with our industry partner Biomerics. Unfortunately, due to their request, I cannot disclose the name of antibody or its target molecule. But you can see that we injected MDA MB231 cells into mouse, and upon tumor development, we started treating them with synthetic antibody or IgG control, and you see there was huge and profound inhibition on tumor development when we used synthetic antibodies against one of the members of F receptors. So with this, I'm going to talk a little bit about how these data and experiences could be used in a comparative oncology approach or for companion animal cancer research. Well, F receptor tyrosine kinases are gaining a lot of attention in human cancer research. And look at this list of possible similar malignancies between companion animals and humans. Investigating the role of F receptors in companion animal malignancies would be very promising. It definitely has benefits to both animals and humans. It could increase collaborations and opportunities for getting research funding and also encourage industry contributions. So this is the area that my lab is focused on right now to basically investigate the role of receptor tyrosine kinases on companion animal malignancies. This is the optimum, workflow. However, if you look at here, we already know some valuable and promising targets from human cancer research, which we could use them in terms of synthetic antibodies or small molecule inhibitors in the context of companion animal cancer research, which is very valuable. So with that, I'm going to the third part of my talk and give you some idea of University of Saskatchewan and the research that is going on there. I'm starting with our Western College of Veterinary Medicine. We are actually the first vet college in Western Canada, including Saskatchewan, Alberta, and British Columbia. We are developing the Oncology Center there, which provides medical and radiation oncology services, and we are also developing Comparative Oncology Research Group there. And we have this Allogeneic imaging suite that opened a few months ago, and we have a full-size PET CT scanner then that is going to be used for both clinical and research purposes. And as a matter of fact, they started using that a few weeks ago, actually. And we have also a rehab center in our future oncology center. There are 4 phases. 2 phases have been already completed. And another news is that we are another Canadian member of Comparative Oncology Trials Consortium as of a few months ago. So about College of Medicine, we have translational cancer research cluster there. Some of the data I showed you with high throughput screening or synthetic lethality we did in collaboration with College of Medicine. We have also Saskatchewan Therapeutic Antibody Resource. They have developed a new synthetic antibody library, which is very valuable for our research and translational work. We have also facilities for genomics and proteomics analysis in College of Medicine. And last but not least, we have Saskatchewan Cancer Center for Cyclotron Sciences. So actually, one of the antibodies that I talked to you is being radio-labeled at this center for use, diagnostic use, or at least research for PET CT imaging. So it's not very clear, but it's a big view of our campus. The point that I wanted to mention here, do you remember this building? Okay. So this is our vet college. The oncology center would be on the west side of it. And here we have the cyclotron, and here is the College of Medicine, Health Sciences, and Medical Hospital, Veterinary Hospital. My point is all these centers are within walking distance from each other and really encourages collaboration between all these centers. And as a researcher in oncology in vet college, I would be very happy to facilitate any collaborations between our colleges and our universities. And thank you so much for your attention.
Thanks. Any questions? Brenda?
So some of the F receptors are also involved in angiogenesis.
Mm-hmm.
And I know that you showed that there wasn't a difference in angiogenesis between your aggressive and non-aggressive tumors, but when you're using these inhibitors, are they going to be targeting, like is it a dual target? Are you targeting the neoplastic cells and also perhaps blood vessels? Do you know that?
I'm sorry, is your question about the antibodies or inhibitors, small molecule inhibitors? Yeah. Like that one that we investigated about the new angiogenesis was specifically about the FP6 receptor that wasn't involved in new tumor.
Cells, right? But when
Yeah.
you treat a tumor with your inhibitors,
Yeah.
whether they're large molecule antibodies or small molecule inhibitors, are you also going to be inducing, in addition to an anti-neoplastic cell effect, an anti-antigenic effect? Have you looked at that?
We haven't looked at that. It's just for inhibitors, again, specifically, we try to use the FDA-approved inhibitors, which are already available. So they have already done some of the testing in terms of toxicity and some other side effects. But for antibodies, synthetic antibodies, definitely that's something we need to look into it in more detail. Yes?
So we do have some, as you know, commercially available certain inhibitors of the axitinib, which has been used in canines, and there are published PK data on that.
Right.
So that would be an easy add-on, an off-the-shelf type of thing. And since we see much more sarcomas in our canine population than in our human population, albeit for the pediatric cancers, I mean, there's some great stuff here that could be applied.
Exactly.
Surrogate.
Yes, no, I agree with you. And it was amazing even with our work, which are not, it was a small part of it. We saw that there are lots of opportunities to develop personalized type of treatment, even for each.
So have you looked at any of the osteosarcoma cases or hemangiosarcoma cases for loss of your ERV6?
Not yet. I started from January, but it's just trying to develop. So, but that's on my list. So we completed designing setting up the assays to look for expression of all these F receptors in different malignancies, canine malignancies. Yes?
So I'm just looking at the phenotype that you have with the cells, so you get increase in cell-cell contact
Yes?
by
Yeah?
increasing the levels of the B6.
Yes, that's true.
And so, and in triple negative, what is can you go back to triple negative? So do they express high or low?
That cell line that I showed you, they completely eliminate FB6. Like the promoter is methylated and no expression of FB6.
Okay. So but and then in the cell, when you re-express it, then you increase cell.
Yes. There. Sorry, go ahead.
So why is it happening? Is it that you are regaining in cadherin or any other cadherin?
Yes, it's a process. It's called epithelial to mesenchymal transition. So I didn't have time to show all the data for that, but it's a big part of our work that is published in Oncology. FB6 actually increases some of those epithelial markers after we restored that. It significantly reduces beta-catenin transcription in the cells and function. And through all of that, it's also physically causes those cell-cell interaction. We did like localization experiments. We saw that FB6 is located at cell-cell contacts area. We stained them with ZO1 and FB6, and they were overlapping. But it's more complicated than that. It's just the physical cell-cell, increasing cell-cell connection. It's reversing that epithelial.
FB6, so you'll get more tumor mass, your growth rate
Yes.
will go up. What about metastatic rates? Intuitively, I would think it would fall off.
Yes, the metastasis, we started to evaluate that. We haven't completed that in that project, but that was on our list. We did some preliminary data. We saw at least microscopically, by counting those metastatic areas, that was a reduction in metastasis. So,
you're
but
more tumor primary, but less metastatic.
exactly, yeah.
That's intuitive.
Yes, no, exactly, because it reverses epithelial to mesenchymal transition. So that process is needed for a tumor invasion and metastasis. However, the effect on the ticks or tumor initiating cell is increasing their proliferation. It's to their disadvantage, because they become more sensitive to
So we can have
treatment.
Michelle cut out the primaries and you cure all the.
Okay, thank you very much.
Thank you so much. It's on.
Do we need, we've got both now? Okay, yeah. Excellent. So we're going to move back into some abstracts now.
Yeah.
Is the slide changes working? And our first speaker is Matthew Chang from the Department of Food Sciences.
Yes. Good afternoon. My name is Matt. I'll be presenting to you some of the major highlights of my PhD project. So, acute myeloid leukemia is this devastating blood cancer that starts with the HSC population shown here. And their role is to produce our body's functional erythrocytes, granulocytes, and thrombocytes. Occasionally, members of the HSC pool can undergo a leukemic transformation to form a leukemic population. At the very apex of this population are the leukemic initiating cells, shown here as the SLICs, and their role is to produce the bulk of the leukemic population, known as the clonogenic blast cells. For the past 40 years, we've been basically reliant on 3+7 induction therapy as our frontline anti-AML regimen. Unfortunately, remission rates tend to vary and also decrease with the more mature patient population. This is primarily because of treatment-related comorbidities and the fact that these compounds just weren't designed to target that initiating cell population that's known for relapse. Recently in our field, several papers have highlighted that fatty acid oxidation is critical to leukemic survival, proliferation, the initiation of relapse, and even the maintenance of chemoresistance. And it's this pathway that's the main method for most normal and malignant cells to break down fats that are less than 16 carbons in length. This pathway starts with fats entering the mitochondria via the CPT system, carnitine palmitoyl transferase system. Once in the mitochondria, the fats are then broken down by different sets of enzymes depending on how long that initial fat was. The end result of one round of fatty acid oxidation is the production of acetyl-CoA for the TCA cycle, NADH for complex 1, and FADH2 for the electron transfer flavor protein. And it's because of that literature I just outlined on the previous slide that inhibitors of this pathway are now being considered a possible chemotherapeutic candidates for AML. My work has been centered around this mixture known as Avocado B. It's a 1-to-1 mixture of two 17-carbon long fatty alcohols, complete with hydroxyl groups at the 1, 2, and 4 positions, and either a terminal double or triple bond. In my early experiments, highlighted here in red, I showed that avocadine, that triply bonded fatty alcohol, was the most potent component of this mixture in both leukemic cell lines and also primary patient samples, all the while sparing the clonogenic growth of the healthy hematopoietic population. We also evaluated the in vivo efficacy of this compound, avocadine, and showed that it significantly reduced primary and secondary engraftment in a patient-derived xenograft study. We also wanted to study the effects of the avocadine compound on mitochondrial respiration. So we turned to high-resolution respirometry. This is an example of one of these experiments shown here. So across the x-axis, you have experiment time going from left to right, and on the y-axis, on the right-hand side, you have oxygen flux represented by the red and gray traces. So after a brief treatment with our compound, we injected intact cells at this point, and that increase in flux that you see there to that plateau is respiration that is coupled to ATP synthesis. It's powered by whatever substrates the cell had access to in cell media. We can also look at maximal respiration over here with an injection of oligomycin and then FCCP. And then lastly, it's customary to end off the experiment with some sort of selective OXPHOS inhibitor. In this case, I chose antimycin A to basically bring respiration back down to zero and prove to you that all the flux you're seeing here is due to the mitochondrial electron transport chain. And the arrows shown up there are basically what I quantified in these bar graphs. And in short, we once again saw that avocadine, that triply bonded fatty alcohol, was the most potent component of this compound in terms of inhibiting leukemic mitochondrial respiration. And because of that, we'll be carrying on with just this compound for the rest of our experiments. We had this hypothesis, though, that this compound wasn't actually inhibiting the electron transport chain. It was actually inhibiting fatty acid oxidation, which lies upstream to OXPHOS. So we had to modify our protocol. You can't really study fatty acid oxidation with whole cells because even if you supply fat to a cell in media, there's no guarantee that your cells are actually using fat exclusively to power the electron transport chain. So to get around this, we had to permeabilize our cells prior to injection, and what this does is it basically strips away all the cytosolic substrates inside the cell that's powering OXPHOS, while leaving all the enzymes and mitochondria and other organelles intact and functional. That's why there's cells in the respirometer from this point to this point, but there's no respiration. It's not until I inject palmitil carnitine, malate, ADP, this cocktail, that we actually see an increase in oxygen flux, which we can exclusively say is due to fatty acid supported respiration, because fat is the only substrate that's in the cell, inside the respirometer with the cells at this moment. So once again, the arrows are basically what I quantified and shown down here. In short, we were able to show that this compound inhibits fatty acid supported mitochondrial respiration. We weren't ready to call this compound a flat out fatty acid oxidation inhibitor just yet. We wanted a more direct measure of fatty acid oxidation, and this led us to a collaboration with Dr. Akhtar from MCB. He kindly ran these experiments for us. We basically supplied a radioactive palmitate to our cancer cells. This gets broken down to radioactive acetyl-CoA, which can then enter the TCA cycle just like normal. Eventually, this acetyl-CoA is liberated as radioactive carbon dioxide. And that release of CO2 is our measure, a more direct measure of fatty acid oxidation. This is more direct compared to respirometry, because respirometry kind of ties FAO activity to oxygen consumption at the electron transport chain. Anyways, we were able to show that this compound actually inhibits fatty acid oxidation in both of our leukemic cell lines. The next question I had was, okay, great, we have this compound that we are pretty sure done in conjunction with some researchers out of the University of Pittsburgh. They specialize in the study of fatty acid oxidation deficiencies in humans. And this experiment is known as acyl carnitine profiling. The gist of this is that whenever you have some sort of deficient activity in any one of these enzymes, there's going to be a massive increase in accumulation of its substrate inside the mitochondria. In an attempt to alleviate this inhibition, the mitochondria will try and have some of this substrate converted to a acyl carnitine intermediate, which can then get pumped out of the mitochondria and eventually out of the cell. This can then be identified and quantified using mass spectrometry. So in our collaborators' field of study, this is commonly done and is a clinically relevant procedure. When a patient comes in with like any one of these FAO deficiencies, they basically are using mass spec to look at what accumulations or depletions in specific metabolites to diagnose whether long, medium, or short chain fatty acid oxidation is being inhibited, and possibly even down to which exact enzyme. So we wanted to apply this same methodology to our project. We used their cells treated with our compound, and basically over here on this slide, WT wild type stands for one of their fibroblast lines that was derived from a healthy donor with functional fatty acid oxidation. We treated this cell line with either a vehicle control or our compound of interest, enzymes associated with long chain fatty acid oxidation. So when we looked at the results, there were over 30 fatty acid intermediates highlighted and identified in this mass spectrometry analysis. Only 5 of them were shown to be significantly different from control to treated groups. So in our discussions with our collaborators, they were telling us, hey, you know, this accumulation of C16 that you have in your cells indicates to you that most likely there's some block at long chain fatty acid oxidation. These enzymes are the ones that process palmitil carnitine and fats of that length. In addition, this significant depletion of acyl carnitines from C10 through to C4 is indicating to us that, you know what, the medium and short chain fatty acid oxidation enzymes are actually probably fully functional. They're not affected by our compound. And that's because this blockage early on in the fatty acid oxidation pathway is preventing replenishment of the medium and short chain fatty acid substrates. With that being said, I hope I've convinced you that this compound avocadine is capable of inhibiting long-chain fatty acid oxidation, which indirectly hinders mitochondrial respiration as well. We'll be continuing to focus our efforts on finding the molecular target of this compound using some of the experiments I outlined here, while focusing on the 4 that our collaborators' experiments highlighted to us. Lastly, I'd like to thank my own lab group and our collaborators. Thanks.
Questions? All right, thank you.
Sorry?
Microphones. I know, I do it every time. It's like you're so desperately going to edit the attention. Can you flip it onto the top of your pants is probably the easiest. You want on each side. Okay. Okay, thank you very much. My name is Adriana. Like Michelle announced, I'm from clinical studies. Today I'll be presenting some results from an owner-reported survey on diet and nutrition in healthy dogs and dogs with cancer that I've been working on. So first of all, a little bit of background information. Studies have shown that most dogs seem to consume a commercial pet food, so about 90%, for half of their diet or more. And dry food was the most popular diet type in this study. And then there were about 50% of dogs that were reported to be fed only dry food. In contrast, about 17% of dogs consumed a non-commercial food for about a quarter of their diet or more. This included things like homemade raw and homemade cooked foods. And then about 3% were eating exclusively homemade food. In cancer, the picture seems to be pretty similar. We see most dogs eating a commercial food, in this case either dry or wet, and then a smaller percentage getting only homemade raw or only homemade cooked, and then slightly larger percentage receiving a combination of commercial and either homemade cooked or homemade raw. However, in cancer, we see a bit of an increase here, with one study citing that 40% of dogs with cancer received a nutritional supplement, and another confirming this with 35% of dogs receiving nutritional supplements. Then 48% of these were found to feed supplements because of the dog's cancer diagnosis. So this brings me to the objectives of my study, to compare supplement use and diet type in dogs with cancer and healthy dogs, and compare sources of information consulted by owners of dogs with cancer and healthy dogs when researching diet and nutrition topics. For hypothesis, I'm expecting number of supplements fed will be higher among dogs with cancer compared to healthy dogs. Dogs with cancer will be less likely to be fed a conventional diet, and owners of dogs with cancer will favor different sources of information compared to owners of healthy dogs. So, the study design, we started with a power calculation to determine our minimum sample size. That was determined to be 102, so 51 in each group, for a significance of 0.05. We designed the survey using Qualtrics and used 52 multiple choice and fill in the blank questions. And then we received our research ethics board approval and began recruitment. For the healthy group, we were recruiting at the OVC Hills Primary Healthcare Center, again, October to February. And then to boost recruitment a little bit in January to February, we did also use social media to recruit. For statistical analysis, we used SPSS, and frequencies were reported for demographic data. Chi-squares and odds ratios were used to analyze categorical data, and then Mann-Whitney U-test for continuous non-parametric data. So this is what my sample ended up looking like. We had a total sample size of 389, so we did exceed that minimum sample size. Owner demographics and dog demographics were similar to other studies that were done in this similar field, so that was good to see. For diet data, first of all, I'll just explain the color scheme here. So from now on, data representing healthy dogs will be in blue and dogs with cancer will be in orange. So we did find that in the frequency data, it seemed like dogs with cancer favored feeding homemade cooked foods and homemade raw foods compared to healthy dogs, whereas the healthy dogs seemed to more commonly feed the conventional dry food compared to dogs with cancer. So we ran the stats on this and found that this was a significant difference. Dogs that were healthy were about 4 times more likely to receive commercial dry food, whereas dogs with cancer were 2 times more likely to be fed a raw food and about 4 times more likely to receive a homemade cooked food compared to compared to healthy dogs. Looking at previous studies done, this is a little bit different. In 2016, we see that the number of dogs fed, or percentage of dogs fed dry food seems to have decreased, and owners seem to be favoring homemade cooked foods and homemade raw foods currently. In terms of sources of information, this is the dogs with cancer. We asked where they look for information when researching nutritional supplements, pet nutrition, and pet health topics. 2 interesting things here. We see the veterinarian is the #1 source of information, so that's good news. However, social media groups was very close second in most of the things we asked about. So again, looking at the stats, we did find that owners of dogs with cancer were about 4 times more likely to consult social media. For the study from 2006, we see the veterinarian is consistently the most popular source of information. So that is, again, good news. Friends and family seems to have increased a bit in my study. And then the internet is definitely something that's starting to increase compared to 2006. Looking at the healthy dogs now, we see some similarities and some differences. Veterinarian is still #1 source of information, so that's great. Social media groups is seemingly decreased in this group. And then we do see pet store seems to be slightly increased. So looking at the statistics, we did find a significant difference here, where odds of consulting pet stores for information about pet health and was about 3 times higher, or 3 times more likely for healthy dogs compared to owners of dogs with cancer. And then about 2 times more likely to consult pet stores for information about pet nutrition and nutritional supplements. So, seems like owners of dogs with cancer and owners of healthy dogs are consulting different information for nutritional supplements in particular. So, next I looked at nutritional supplement differences between these groups. So, in the group of dogs that do not have cancer, of 257 respondents, 110 reported feeding nutritional supplements. So that's 43%. In the group of dogs that do have cancer, of 132, we saw 86 report feeding nutritional supplements, so about 65%. This difference was found to be significant. Owners of dogs with cancer were significantly more likely to feed nutritional supplements. Then when we look at the range data, this is the number of supplements fed per dog. So in both groups, the minimum number of supplements fed was 1, which makes sense. And then the maximum was higher in the dogs with cancer. So we see a range of 14 versus a range of 19. And the mean number of supplements fed was also increased in the dogs with cancer. So this difference was also found to be significant. Dogs with cancer were receiving more nutritional supplements than dogs that do not have cancer. So this next led me to look at the types of supplements being fed. This is a graph of the total reported supplements, all grouped into similar categories between dogs with cancer and healthy dogs. So there are definitely some trends among the 2 groups here. When I ran the statistics on this, I decided to look at some of the most popular supplements fed between the 2 groups and see if there were any significant differences. So over here, we have the supplements that were found to be significant. CBD oil, turmeric and curcumin, mushroom supplements, and supplements containing a multitude of different compounds that was marketed as an immune-boosting supplement were all more likely to be fed to dogs with cancer compared to healthy dogs. Supplements like fish oil, omega-3s, multivitamins, multiminerals, probiotics, and glucosaminochondroitin were all popular among both groups, but there weren't any significant differences between healthy dogs and dogs with cancer there. In terms of previous studies, here we have the study from 2016 again. These are a few of the supplements that were similar in both studies, and there are some differences and some similarities, but I think most prominent here is herbal supplements seem to be on the rise compared to that study. So before I wrap up, I'll just revisit my hypotheses. Number of supplements fed were higher among dogs with cancer compared to healthy dogs, though the frequency data did indicate that dogs with cancer were more commonly being fed dry food. We also did find that dogs with cancer were more likely to be fed a homemade raw or a homemade cooked diet when compared to healthy dogs. And then owners of dogs with cancer and owners of healthy dogs were both using the veterinarian as their primary source of information, but we did find that social media was favored in the healthy dog, or in the cancer dogs compared to the dogs with cancer. So for future directions, definitely more nutritional research is needed in these supplements, especially research pertaining directly to dogs. We don't really know a lot about the safety and the efficacy of some of these supplements. And definitely client and clinician communication and education when it comes to consulting on online sources and making sure that they're using reputable sources of information when making their dietary decisions. There's a couple acknowledgements here of the people who made this study possible, some of the references that I used throughout this presentation, and I'll take any questions now. Yes.
I was wondering if you reported the ages of the dogs in the study. So the dogs with cancer be a much higher age group, which might explain some of the different feeding regimens?
Yes, so I did do that. I think it's in one of my supplemental slides here. Bear with me. All right, so here are the ages. This is also compared to that 2016 study. So dogs with cancer were slightly older than the healthy dogs, so definitely could be a conflict in some of the supplements that we saw fed. Yeah.
So you think these results may reflect the type of owners that live in well and around? It's like the demographics, the way people look at things, the way people get information.
Yeah.
Compared to just looking at this small population in Ontario to, let's say, an international study that had been done on the same.
Yeah, great question. In the end, we weren't actually very successful in recruiting at the cancer center, so most of the dogs in the survey were from different areas. I have the specifics on that a little further back here. Oh, maybe not. So we didn't get too many from Guelph. Most of the respondents were actually from the United States, and then Canada was the second, so seems like it's a bit of a more broad picture there.
Any other questions? Great, thank you.
Okay.
So we're going to take a short break.